Okay. Yeah.
This is the second speaker of our morning session. It's a pleasure to welcome Evgeny Tsibatse. He's a research scientist in the Netherlands Organization for Applied Scientific Research, and his research interests include machine learning and its application to systems biology, medicine, automating reasoning, and information retrieval. His background is in mathematics and computer science, and then he went on to do a PhD in statistics. And today he will be talking about machine learning for modeling microbiota composition. Welcome.
Thank you very much. Thank you for intro. So what I would like to talk about is a couple of techniques that we have recently developed for modeling early respiratory microbiota. And for those of you, from biology side or medical sciences side, let me just say a couple of words about machine learning itself. So it's a recently emerging kind of field on a cross border between statistics, computer science, and math. And it's concerned about developing type of techniques which are applicable to, let's say, high-dimensional data sets. And many problems, if we are looking at metagenomic data sets, they're quite high-dimensional. So it seems like these type of techniques can have a nice application in that area. Let me say a couple of words about the project I would like to describe. So this is a collaboration with University Medical Center of Utrecht and some people from Amsterdam Medical Center and also University of Liverpool. So what we were concerned with in this case was trying to look at early respiratory microbiota compositions. And let me motivate why it is actually an important problem to look at. Okay. So basically from nasopharyngeal niche, pathogens, they can basically spread and cause diseases such as, otitis media, pneumonia, sepsis, and meningitis. And the thing is, in a recent United Nations report, actually the respiratory infections are considered to be #1 killer of the infants and babies. And if we can understand a bit better what is the microbial composition, in the nasopharyngeal area, and perhaps model it and understand its relationship with the, disease, let's say it that way, perhaps we can actually do something about it and maybe even save the human life, So I'm working with many people who have a microbiology background or systems biology background, and what they are telling me, as a simple guy coming from the math side, they always look at the complex communities. So it's really the ecosystem, say, that determines, what kind of species they are and their dynamics. In fact, these ecosystems, they could be actually quite beneficial to the host because they can stimulate immune system. Also, they could be quite good for metabolic function. Now, the thing is, the question which these medical doctors are really interested in is it the case that some particular community could be, for example, susceptible to the disease or to the health? So they would like to just see, whether or not perhaps if they understand that there is some particular type of ecosystem which is associated with the less respiratory infection, right? Could we then perhaps develop some kind of probiotic or change the development of that ecosystem in such a way that we minimize the amount of respiratory infections in the infants, for example, without giving them antibiotics, which is a big thing. Okay, so for that purpose, actually, there was quite a large study conducted, and this study's name is MINUS. So about 1,000 kids were actually sampled. So the nasopharyngeal swab was taken, and these samples were sequenced. So we have a 16S kind of a data. And from these 1,000 kids, 60 kids were actually sampled at 6 weeks, 6 months, 12 months, and 24 months time periods. And for these 60 kids, we actually have an additional metadata. And this metadata looks like that. So in fact, we know whether or not some particular children were, whether they were breastfeeded or whether or not they have the cold, whether or not they were attending the daycare, etc., etc. Also, we know something about the respiratory infections there. And well, the natural thing, what we would like to do, and basically many practitioners who would like to look at such a data set, they use a tool like a hierarchical clustering. Well, this thing is called a unifrac. Perhaps you know it quite well. What you see here is just some kind of a plot, a hierarchical clustering plot, where every bar indicates an individual. So it's a microbial profile of the child. So you also see here that, for example, some of the most abundant species are species like Moraxella, strep, Corynebacteria, etc., etc., etc. So it gives you kind of a feeling, okay, so what is happening there in your data set. However, well, yesterday I think there were also a couple of talks and discussions about the phylogeny and how important it is to be, how important phylogeny is for example, for measuring the similarity between the individuals. And to be honest, I would like to be a bit conservative here because, so as you see, for example, here, there is one big cluster that contains all these, kids who are dominated by Moraxella. But the thing is, we actually pretty much know, or at least that's what microbiologists are telling me, that if the species are phylogenetically close, it doesn't necessarily mean that their function is kind of that they're doing similar thing in your organism. I mean, think about E. coli. There were some outbreaks of E. coli, and some E. coli in Germany, and some E. coli are actually harmless. So phylogenetically, if you do this type of analysis, they will really go in one cluster, in fact. And yeah.
So here, this is so the leaves are the data sets and not the organisms in this.
Yeah, these are individuals. So every symbol is an individual, and it's a microbial profile dominated by some particular mark. So
what is the tree on the inside? It seems to be connecting the individuals somehow.
this. So it's just a hierarchical clustering tree.
Oh, is that?
Nothing more. It's just a visualization. Right? So so yeah, it's a clustering not on a species. It's a clustering on the individuals. Okay. So the features are actually here the OTUs. So basically OTUs, which belong to the different. So that could be Moraxella number 1. In some cases, you actually don't even know what kind of Moraxella it is. So it's just a Moraxella OTU. So frequently, actually, people don't even bother, thinking about type of things, how, similarity actually affects your grouping that you have, or what is the true number of clusters there, or what is perhaps the most appropriate technique to do this analysis. And I think these are quite fundamental questions, and they're pretty much, hot topic in our field. That Shay Ben David, he works on theoretical foundations of the clustering techniques. And I think it's, at least if we, it's quite important at least to keep some of these questions in mind when we are doing such an analysis. So what we try to do is, we try to look at this data set and perhaps think of what are the most appropriate techniques which we could use, and also adapt these techniques appropriately to see whether or not we can recover, some important information or understand what is happening there in the data. So one of the technique which is, I don't know, quite popular, I would say, in machine learning, is so-called a spectral clustering algorithm. This is actually quite a simple technique, which was introduced by Andrew Ng and Mike Jordan somewhere around 2002 or something. After that, different variations of this algorithm have been also widely applied in different journals, and etc. The idea is actually quite simple. So one of the first steps that you have to do, you have to construct so-called Laplacian matrix of your data set. And that's by, you have to compute adjacency matrix of your data, and that's by using some similarity measure, let's say Gaussian kernel or something like that. Then you compute the Laplacian of it, then you do the eigen decomposition, and you look at the eigenvalues of that Laplacian matrix. So why would you want to do that? In fact, there are a couple of papers, theoretical papers, that also show to you that, that also indicate that basically Laplacian matrix of that particular data set would capture the intrinsic properties of the data, basically the manifold. So if you look at that upper plot with the three rings up there, you know, if you apply a k-means or I don't know, some mixture, Gaussian mixture or something like that to that type of two-dimensional data set, you will get a horrible result. So you will never be able to recover these rings. Well, this technique actually can work pretty nicely in this type of setting when you assume that there is some kind of underlying manifold in your data. So, and actually it works also quite well on high-dimensional data sets. In fact, this is just in 2 dimensions, but well, for high-dimensional data sets it's quite nice as well. What we did, we actually extended that technique and by using so-called a co-regularization analysis. And you know, I wouldn't bother you now with matrix analysis and some linear algebra here, derivations, but I would like to convey to you the idea behind it. So it's based on the idea of co-training approach, and that's a paper by Bloom and Tom Mitchell. The idea is quite simple. So what they were concerned with was looking at classifying the web documents, okay? And they looked at the web document via 2 views, 2 data representations. One based on the text that this web document contains, and another based on the links that the web document contains. So what they wanted to do is they wanted just to build a classifier that, given a document, predicts whether or not the document belongs to a news category or sport category, something like that. And so now they have 2 classifiers, each of them trained on different views, one based on the text that the document contains and another based on the links. But in addition to these 2 classifiers, they introduce so-called a co-training term, and that term penalizes your prediction function when there is a disagreement between these classifiers. So basically what it does is that it always promotes an agreement between these different hypotheses, all right? And what they've shown also empirically, and also there's some theoretical justification to that, so you get a sharper mistake bounds, and these type of techniques actually work quite nice. So they are kind of ensemble techniques, but not just an ensemble techniques. So they have this additional term that promotes the agreement. What we did basically, we extended that idea to be applicable to this spectral clustering. And well, some details are published in our paper on PLOS Computational Biology and follow-ups. But the idea here is that basically the way how you construct the adjacency matrix of your graph could be done via different type of similarities, and you basically can incorporate that information into account and get a bit more stable results. Okay. Let me say one more thing, and then I will switch to the results, perhaps the most interesting part. So that was about the technique, and another thing is, for example, how to determine the groups in your data. And well, what many people do, they use a silhouette index or L-curves or stuff like that. So there is a technique which I think is pretty much appropriate, and perhaps you heard about that. It's so-called the consensus analysis based on the co-occurrence matrices. So the idea is extremely simple. So once you have your data set and you want to estimate what is the true number of groups, instead of just randomly cutting that hierarchical tree into classes or predefining the number of clusters that you have for the k-means or whatever, what you do, you actually look at the range. So you say, first initially I'm looking for, let's say, 2 groups, and then you see how frequently 2 individuals or 2 subjects co-occur together when you predefine 2, 3, 4, 5 or 10 clusters or whatever. And then you just compute how often the 2 objects co-occur in all these different simulations. And you construct the matrix, which is of dimensions your examples times examples. So it's a symmetric matrix. On the diagonal you actually have ones, because the same object person co-occurs with himself always. But the nice thing of this is that the groups actually naturally emerge from your data while you predefine all different solutions, right? So it's basically, think of it as some kind of averaging, or averaging over all different number of clusters that you have, and then you get such a plot. So this thing also, well, the way how these type of techniques are evaluated and how we also evaluated our technique was that you pick up the data set, which actually has the label information. Let's say some multi-class classification data set. You strip off the labels, and then you apply this type of technique, and you see whether or not you're able actually to recover the groups, okay? And there are specific, well, measures of accuracy, how good your clustering technique is. Okay, so what we did, so I hope that I've managed to convey the message about the fact that you know, we try to kind of pick up a good technique out there, adapt it appropriately towards our problem, and see what we can get out of it. So one of the things which you can notice here is the following. If in the previous, well, I don't know, in the previous plot there was one big green group of all these Moraxella species, all right? What you see here is that there clearly another group of the Moraxella C, Moraxella 1, Moraxella 2 is appearing as a separate cluster out there. Furthermore, you also see, well, there are some particular type of Corina bacteria which are known to be associated with the breastfeeding. Okay, they are also going in a separate groups. But so what is happening here is that we apply this technique on the data set at every single time point and see what are the groups and what is happening there, what is the profile of this kid. But in reality, this is not precisely what we would want to do. Yes, we would want to understand and describe, you know, these microbial profiles, but in fact we would also like to take the time resolution into account. And what we did was actually pretty much simple stuff, what people also frequently do in time series analysis. We simply look at the relative differences, okay? Relative differences in abundances per every OTU between the different time points. And we look at the consecutive time points. Let's say time points 6 weeks to 6 months, 6 months, 12 months, 12 months, 24 months. So instead of having these 4 separate data sets, now we have 3 data sets, okay, where every child is represented as a long vector of the relative abundances, of percentages of relative abundances. So increase, let's say 50% in OTU Moraxella between the time point 1 and 2, and decrease in strep, and etc., etc. Once you do that, actually what you can further do is just to compute this L2 norm, which is just the length of that vector. What it shows is, you see that, for example, if you look at the 6 weeks to 6 months, 6 weeks to 6 months interval, you see that this area, okay, this is the blue part, is somewhat larger than the rest. So when we showed this really simple plot to our collaborators, they told us, we always knew that when the child is born, they are reasonably sterile, okay? They don't have their own microbial profile. So we were actually suspecting that, in fact, during this early period of time, the child gets his own kind of, in quotes, microbiome, okay? And you would expect that during this period of time, the development or the changes in microbial composition would be the largest. So they were kind of suggesting that, but they were saying that, well, we never could prove that. So to some degree, this is an indication that actually occurs. But so this is concerning the change. But now we could also do the same thing, but now applying our technique on these data sets of change. So what you see here, you see these clusters, right? So these red blocks. But now these are clusters of the kids who have their microbial profile changed similarly between the different time points. Okay? So, and what you see here is that early in the beginning of their life, you know, that they're kind of having a similar pattern, but then towards the end, so between the 12 and 24 months, what you actually see is that they become quite diverse. And also the explanation of that thing is that, you know, as the children grow, they go to the daycare, they also interact more with the friends, and it's also kind of a natural that you have more variety there, so that they start really developing in a different way. Okay. Furthermore, if you actually look at what are the really drivers of these different groups, you see that in some cases these different Moraxella species, for example, they increase. But in the other case, between let's say 12 and 24 months, you see that in one case, one Moraxella is kind of increasing, but while the other one is decreasing, and the vice versa. So this is all nice, but then what we wanted to see is what do we get? What kind of a message we actually get out of this? And we did this simple plot to understand what's happening. So let me try to explain to you what's going on here. So these are the different time points, 6 weeks, 6 months, 12 months, 24 months. And you see there that for 6 weeks there are, a couple of most important species. In fact, these are not the most abundant species, but we run some feed-forward marker selection techniques, which I haven't really explained. But so you can pick up kind of biomarkers, okay, per every time point. And what you see is that if the child at early stage has a Moraxella profile, okay, so he is dominated by Moraxella, he tends to stay in that Moraxella profile throughout this 24 months period of time, okay. Furthermore, on the other hand, so if you are dominated by strep, okay, you tend to, in some cases you do go to Moraxella, but actually you can also end up in this part. Now what we did further, we just very simply calculated how often, the people who are, these kids who have this Moraxella profile also have the respiratory infections. And in fact, more than 85% of these kids, they never encountered a respiratory infection. So there was a paper also published about a zebrafish. And what they were trying to do there is they were trying to inoculate the zebrafish at very early stage with some particular microbial profile, and see whether or not that profile actually keeps on staying as the zebrafish grows on. So what we think, or what our hypothesis is, how we try to interpret it, is that perhaps a similar phenomenon also occurs in kids. So what would be nice if we could somehow try to shift these individuals who are dominated by other type of species towards a bit more healthier profile, okay? So what I also have to say, you have to take this thing with a bit of a grain of salt. Of course, it's never the fact that we are not talking about causal relationship here. So by moving, let's say, by changing, by giving some kind of probiotics to a child, and by shifting his microbial ecosystem, let's say, looking to be more Moraxella-like, and we think that this is perhaps a healthier kind of ecosystem, it doesn't necessarily mean that he would encounter less respiratory infections. But there is a growing amount of studies, also published in nice journals like New England, where they were doing a fecal transplantation, and they were really showing that actually by changing microbial ecosystem, you can also change the health status. Okay, so let me then go to the conclusions, and these are pretty much kind of general conclusions which I wanted to point out. So what we tried to do is we had an interesting data set, a metagenome data set. So we tried to really develop and apply then appropriate technique which we think would allow us to understand what's happening there in a bit more details, rather than just using some standard tool or standard technique there. So I think given exponential growth of the data, these type of techniques which can really handle very high dimensional data sets would become more and more important. And I've seen that, many people already here, they're using the L1 kind of SVMs and et cetera, et cetera. So I think these are really nice models which can be even further improved and applied for particular problems in metagenomics. And and to me, as computer scientist and mathematician, I think that usually by improving the model, you actually get better prediction, better markers, and better application at the end. So that's all I want to say.
Thank you. Is there any question?
I just wanted to go back to that tree that you showed, where the leaves were the data sets. I don't understand at the very, very beginning.
The unifrac?
Yeah, I don't.
Okay.
Why? Yeah, but why would you want to do a hierarchical clustering of those data sets? I mean, why would you show the tree? What information is in the tree? Why would there be a tree-like
Okay.
structure on a bunch of data sets? They're not evolution. There's no evolutionary relationship between the different kids. What information is in the tree?
May I say something? So the thing is as follows. So you would like to look how the microbial profile of how two kids are similar to each other.
Yeah.
We look at the kid based on his microbial profile. Okay. So if I have a microbial profile which is similar to yours, we will be close in this tree. Now the thing is,
Data is tree-like, but there's no reason why it would be tree-like.
okay, well you can use a k-means, whatever you like. So the thing,
that was my question. Why are you?
why tree?
Or just
Well, the
you
thing
actually
is,
look at the tree
here you're using a phylogenetic distance as the measure.
They're not valid. They're not. I mean, that's my point. You said it, phylogenetic distance, but they're not valid. I mean, the individuals are not different.
No, but species are. So the way the species are, right?
Yeah, but the
So-
reason why these different individuals have different species has got nothing to do with phylogeny. It's just
No, but you measure similarity based on phylogeny.
This is not a phylogenetic tree.
I know. Yeah, that's why I'm asking why use a tree at all, because it's not the best.
You can see differences at many different scales. It's way better than PCA.
Yeah.
Okay.
Is there any other question? Candy?
So I have a similar question to what I asked last time. So later on in your talk, you have you're showing how some bacterial species are sort of staying constant or dropping. Can you go forward to that? And when you talked about this, keep
This?
going. No, not that one. Keep going. Keep going. That one. So these are not the dominant,
No,
right? And yet
some of them are, some of them are not.
right. But you were really interested in looking at this sort of computing this graph. So to me, this means you're really interested in looking at the relative abundance of these bacteria, right? In the metagenomes of the people you're looking at.
Well, it's a simplification, yes, but from the practical perspective, there are more species there, of course, so there are more OTUs. These are
Right.
just some which.
But you're looking specifically at these, and the question I'm trying to get at is your ability to estimate the relative abundance of these things is impacted by how well the methods you're using are able to estimate those. So the question is, how abundant are these things that you're measuring? And how are you assessing the ability of your methods to estimate those relative abundances of things at this level?
Yeah. Well, I think so. It's a difficult question, definitely. But what people usually do, they look at the mock samples, for example, right? So you take a look whether or not there is some contamination going on, and that's what your question is, right? Okay.
I guess I'll ask you later.
All right, but it's definitely so. The relative abundance is definitely important in this case, right? So it's and if the methods, if in your PCR or amplification, you're not getting a proper relative abundance levels, this probably will not work also. Yeah. Okay.
Right.
Yeah.
I have a question. So you showed us how these co-occurrence analysis can work to find out clusters. So
Yeah.
is it, and it sounds, I know that you didn't go into the details of
Yeah.
Is it simple to implement with,
Very simple.
so it's just like counting the number of times?
So well, I have all the code, so it's also I can send it to you. It's publicly available. It's really simple.
Yeah, I understood that, but I didn't quite see what you were doing with that when you were looking at this relative abundance. Was this being applied also in the
Yeah.
tree?
Yeah.
I didn't quite see how you did
So,
that.
so the thing is, these clusters which you see here, and for example, well, take a look at these plots. So these are dead consensus matrices. So It's a technique, so we have a technique, let's say the spectral clustering or k-means, whatever you want, okay? And then you apply it to your data set. Now this data set is not just the data set of relative abundances, but here we are really looking at changes in the microbiome, right? So if you do remember that point. But okay, never mind. So you just apply the technique and you run it in multiple simulations with the various ranges of the clusters that you allow. And that's what kind of plots you get.
So you don't actually show which of the clusters that you choose.
Well, so yeah, it's a good point. So the clusters are, of course, these ones that are the block ones. So we have a procedure which can automatically extract. It's a matrix decomposition. Okay, the red blocks from this matrix. It's not a picture. I mean, it's a matrix, right? And it's just arranged in such a way that you have this visualization, these blocks, kind of nicely aligned. But in reality, it's just a matrix that contains information on the co-occurrence between different subjects. That's it.
So, yeah.
Oh, okay, maybe.
But you,
Okay.
so you view choosing the number of clusters as a...
It's a variable. So I choose from 2 to 20. So I look at all different combinations of clusters. Okay. I predefine them. I run my technique, and then I compute how often these two individuals co-occurred in the same cluster. Okay, over all these 20 runs.
Okay, but you view the variation between runs as being statistical in some sense.
What do you mean? Yeah.
To indicate uncertainty
To indicate uncertainty.
in a
Yeah.
clustering.
No, I mean the only. So it's not for uncertainty. This is just for estimating number of clusters there.
But that is a... What does number of clusters mean? I mean, everyone is different.
So, okay.
If you look carefully enough.
Well, what does a number of clusters mean? It's a number of groups that you have there, right? So number of groups of individuals that are similar to each other based on their microbiological profile.
Yeah, but there are many different levels of similarity depending on.
Well,
your
if you also pose it in such a perspective that then everybody is unique to each other, right? Even if really tiny changes in microbial abundance of if some OTUs can make you different from me, yes, then every single individual goes into a separate cluster. That's the point you're trying to make, right?
Well, so at some point you're making a choice about how similar things have to be in order to cluster them.
Yeah.
And I want you to be clear about
No, that's just a range of the clusters that I predefine, nothing more. So, and I say that's from, I don't know, 2 to 20. And in fact, if you say from 2 to 10, this plot would look pretty much as similar. That's the only thing I'm trying to say. Yes?
Wally?
So just a further question for clarification. So the clustering is done. So the clustering, that's the thing, is done. First thing you do, you get your relative abundance, which you define as a sort of the difference between two time points.
Yeah.
So you get that for each kid,
Yeah.
and you divide by the total or something to make it relative. Yeah.
No. So there is definition of relative change. It's a time point past minus time point before divided by time point before abundance. That's it. I do that for every OTU.
Yeah. And then you basically run some form of cluster analysis.
Yeah, on top of that.
Which could be more or less anything you choose.
Yeah.
A whole variety of different things. You chose one particular way to do it. That's okay.
I can
And then
justify that.
Sorry?
I can justify that. So in fact, we can also show that this particular technique. So we did comparison of multiple clustering techniques. And in fact, there are a number of papers in BMC bioinformatics, in PLOS computational biology, that indicate that there are some type of techniques which are empirically shown to work better on metagenomic data sets. Furthermore, you also have a theoretical justification why to use some technique under certain conditions.
And the particular technique you used, I didn't understand.
Spectral clustering.
I don't understand what that is.
Oh, you don't understand it? Oh, I can explain it one more time. Would you like that?
Please do that.
Okay. So the idea is quite simple, at least That's how I see it. All right. So imagine you have a data set, and well, a data set, imagine at this point is just a kid. It's a long vector of OTUs, okay, so relative abundance of particular species. So what you want to do is that you want to compute some kind of a graph, or you want to compute the adjacency matrix of the graph. And this adjacency matrix, weighted adjacency matrix, may depend on any similarity measure, let's say Gaussian kernel or something like this, okay? So you compute the similarity, you compute this adjacency matrix of the graph. After that, you actually compute the Laplacian matrix of it. There is a reason why you're doing that.
Could you explain what that means?
Laplacian matrix?
Yeah.
Well, Laplacian matrix is a degree matrix minus the adjacency matrix of the graph. It's a definition from the graph theory. Basically, the interpretation is that it tells you something about, well, how the groups of the connected nodes there in the graph, okay? Once you do then you do eigendecomposition on that matrix. And what you get is a couple of first eigenvectors. Now these eigenvectors also tell you something about the structure of disjoint components in this graph. Okay? So basically you don't necessarily need to look at, it's like, think of it as a data projection, but in a clever way, where the manifold structure is preserved. See? So there is a really nice paper, if you'd like to look at that, by Andrew Ng and Mike Jordan. And then there are also some nice explanations and justifications why this is nice. And like I was mentioning before, so if your data set really has a manifold structure, this is genuinely the thing to do, in my opinion. Okay?
Is there any other question? Okay, so thank you very much.
Thank you.